Okay, about 30 more seconds, a little bit less. Okay, let's go ahead and transfer over to our class kick. Thank you all for letting me know how you're feeling. I hope that you're excited to get back into some practice. Let's get into the do now. If you recognize this, this is question 19 from your exam. You were given a code. Which of the following polypeptides is coded for by the mRNA sequence? So here's what you do. I'm going to go ahead and zoom out a little bit. We have AUG, GUU, AAA, CGA, CAA, UCC. This is going to be our mRNA. I'm going to drag it down here so we can make this easy. So AUG, I hope that you remember. That's the only one that's ever going to code for methionine. GUU, you go to GUU, valine. AAA, lysine. CGA, arginine. CAA, glutamine. I was thinking glue. And UCC, serine. Okay, now, if you did this, congratulations, you wasted your time, right? Now, we are given multiple choice tests, right? Remember, use the test. Use the test against itself. We know that AUG is a start codon, right? It's only going to code for methionine. So right off the bat, you should get rid of A and D. Like, you shouldn't even look at these things. Look at those disgusting things that don't have methionine starting off, right? Gross. Now, if you wanted to play some 3D chess, you also notice that B and C has different numbers of codons. So hypothetically, you could just count. You could just go 1, 2, 3, 4, 5, 6, get rid of C, and then go with B. 1, 2.
Wait, sir.
Oh, wait, I'm sorry. You're right. You're right. Sorry. You get rid of B, you go with C. Now, what you could also do if you didn't want to go through that work of counting your codons, if you just translated the first two, methionine and valine, boom, you're done. Because you can see that asparagine is already different, right? Or aspartic acid, whatever that is. So, easy question, right? Now, there's giving you this big long sequence. You could take your time and double check and make sure that you get it all correct. Or you can use the test, use your time wisely, get rid of the answer choices that are dumb, right? So again, methionine was our first, so you get rid of anything that doesn't start with methionine, translate that second one, and then boom, you're done. That's it. And just like you saw with me, never get cocky. When you get cocky, you can make mistakes. So it is good to check your work. So thank you all for double checking me. Okay, any questions about that? Great. Let's go ahead and get into it. Here's our objective. If you hadn't had time to get that down yet, we're reviewing our Unit 4A concepts and using Punnett squares to analyze patterns of inheritance. I like this picture because you can see how even though it has the recessive allele, it's not expressed because of that dominant trait. So I like that. That was cool. Okay, let's zoom back in. Boop. For each of the genotypes below, determine what the phenotype would be. Purple flowers are dominant to white flowers. So we're talking about phenotypes. We're talking about the physical characteristics. So can someone tell me what you'd expect to see for big P, big P?
Wouldn't it be 100% purple flowers?
That's true. So this is a homozygous dominant. We got two dominant traits. The dominant trait says is purple. So there you go. That's purple. You know what? Let's be fun. Let's make this purple. Let's go for it. Look at that. Oh man, it's great. Purple. Now here we have a heterozygote. What that means is we have one capital letter, one lowercase letter. What trait is going to be expressed with one dominant and one recessive allele? Your choices being purple or white. Brighton says purple. Brighton, you are correct. So again, whether you're homozygous dominant or heterozygote, as long as you have one of those dominant alleles, you're going to express that dominant trait. Now, little p, that's going to be double recessive. So that means that it's going to express the recessive allele. In this case, it's going to be white. And since I can't do white on white, I'm going to make it a black background. Isn't that fun? Look at how much fun we are having. It is incredible. There you go. Okay. Next, hairy knuckles are dominant to non-hairy knuckles. And believe you me, that is true. I'm getting there. I'm not quite there yet, but I'll get there soon. My dad has pretty hairy knuckles. Okay, so hairy knuckles are dominant to non-hairy knuckles. So if we're dealing with a homozygous dominant, that means that we are going to have very good. Hairy knuckles. And just like what we saw with the purple, even if you have a recessive allele, you must have two recessive alleles to express it. So big H, little h, you still have that dominant trait, so that means that you're going to have them hairy knuckles. Yep. And last but not least, we got the recessive. We got two recessive alleles, which means the recessive allele will be expressed, and that's pretty much going to be non-hairy. There you go. Now, bobtails and cats are recessive. Normal tails are dominant. So going off that, we can see here, little t, that's going to be our recessive. So we'll say that's bobtail. Everything else then, normal. Normal traits are dominant. So our homozygous dominant, our heterozygote, both going to be normal. Now, in the science world, we don't like to say normal because it makes some people feel bad. So what we say is normative, which, you know, not much better, but you get the idea. Okay, so for each of the following, write whether it is homozygous dominant, heterozygous, or homozygous recessive. So here, capital A, capital A. What is that going to be? We have two capital letters. Remember that homo means same. If we have capital letters, that means that we're dealing with a dominant trait. So capital A, that's going to be a homozygous dominant. A little bit bigger. There we go. Now look to the left, to the right. We've got little g, little g. Again, we have two of the same. That's going to mean this is homozygous recessive because we've got two lowercase letters. All right, we got a big P, a little p. Is that going to be homozygous or heterozygous? Is that going to be homozygous or heterozygous?
Heterozygous?
Very good, Josiah. Thank you. So we got a capital, we got a lowercase. That's going to make it heterozygous. You'll notice right next to it, exactly the same thing. We got a capital I, we got a lowercase I. That's going to be heterozygous. Remember that hetero means different. We got different capitalizations. It's going to be heterozygous. Little t looks very similar to little g, right? So we have homozygous recessive. Big T looks very similar to that big A, right? So that's going to be homozygous dominant. And there we go. Those are our alleles and our genotypes. Now, it's important to remember that here we see only one letter, right? We have either A's or G's or P's or I's or T's, right? So you're not mixing letters, not yet. We're still only dealing with the one trait. So you have one trait, you're going to have one letter. Now, when we get to two traits, then you'll have two letters, like A and T or whatever. But for now, we're dealing with, your capital and lowercase are going to be the same letter, just different capitalization. So I'm going to write that down here. I'm going to make it red. So, that says ohm. One trait equals one letter with two, let's say, one letter uppercase or lowercase. So we're not dealing with two different letters, we're dealing with two different capitalizations. One uppercase, one lowercase. I noticed this on some of y'all's class kicks yesterday when you were doing your heterozygotes. Some of y'all are mixing up letters. It's just going to be one letter at this point. It'll get more complicated later. Don't you worry about that. But for now, that's where we are. Oh, I was going to take a picture. So let me take a picture of this. And we go. Putting that into the chat box. Okay. Boop. Our first practice problem for the day. Now you notice this black bar up here, it's going to be constant for the next three. We're dealing with the same alleles for the next three practice problems, just to keep it simple. So in dogs, the gene for fur color has two alleles. The dominant allele, capital F, codes for gray fur. The recessive allele, lowercase f, codes for black fur. So gray dominant, black recessive. The female dog is heterozygous. The male dog is homozygous recessive. Figure out the percentage or ratio of possible phenotypes and genotypes of their puppies by using a Punnett square. So mom is heterozygote. So that makes it easy, right? We're dealing with F's, so we're going to have big F and little f, right? There we go. Now, the male dog is homozygous recessive. That means that he is going to be little f, little f. So you bring it down, you bring it over, you're going to have big F, little f, little f, little f, big F, little f, and little f. Oof, those got smaller and smaller. Okay, I'm going to switch over to text because that's just easier. So as far as big F, how many of those did we see? We saw none of those. So we got 0% of those. If it comes to big F, little f, I saw there were 2 out of 4. Whoa, it's actually going to be too big. We're back small again. That's going to equal 1/2. And that's going to equal 50%. There we go. Now, little f, we see the exact same thing. We see 2 out of 4. That's going to equal 1/2. That's going to equal 50%. So 50-50. If they want to be real mean, they could turn this into a math question and say, if given a litter of 30 puppies, how many of the puppies are going to be black, right? So percent black fur, percent gray fur, it's going to be the exact same thing we saw here. Control V. Let's make it red. There we go. Whoops, missed a couple. I don't know why it doesn't want to participate. There we go. 50-50, right? So if you're given 30 pups, that would mean that 15 of them would be black, 15 would be gray. Now, in real life, what you would probably more see if you had 30 pups, you would say that, 16 would be black and 14 would be gray. Like you would see like you're almost 50-50 because there's other factors at play when it comes to not only fur, but all of your genes. There's a lot of interplay. I was talking with Eileen yesterday. There's environmental factors that play a role in the type of genes that are going to be expressed. So realistically, the numbers are not necessarily going to be that clean, but you will see a very close number. So like not exactly 50-50, but pretty close. Okay, let me take a picture of this. Put that into the chat box. Okay, so for this next one, I'm going to give you 3 minutes, and you are going to solve this one. Okay, so 3 minutes on the clock. Time starts now. I'll put on some music. Oh, wait, I can't put on music because then I'll get copyright strikes. Oh, never mind. We'll just have to sit here in silence. Hello, Ayla. Hello, Eva. Hey, Jay. Hi, Kaden. Hello, Natalie. Oh, Dolly, good to see you. Kinda. Because I'm not really seeing any of you, right? I'm seeing circles with letters in them. About a minute and a half left. Okay, about a minute. And 30 seconds. I'm going to start writing some stuff down. Okay. So here's what you should have for the parent genotypes. We're told that both the female and male dog have black fur. That means that both of them are going to be homozygous recessive. So you got two little f's, two little f's. So then you go through here, you do your, I don't know if I can do a text box. I should be able to do it, right? That would mean you have little f, little f, little f, little f, little f, little f, and little f, right? So you don't have any big F. You don't have any big F, little f, right? Because there are no dominant alleles here. So that means that 100% would be black. Now, like I said, when it comes to real life, it wouldn't necessarily be 100%. Now, and I'm not saying it couldn't happen. It's just not super likely. What you might notice is that if there was a litter of, 100 puppies, then 98 or 97 would be black. And the other ones would have, mutations or maybe patches. It just kind of depends. There we go. Pretty simple, right? This one, not too hard. No dominant alleles, so we don't see any of those. Okay? Same thing on this one. I'm going to give you, let's go 4 minutes on this one. It's a little bit more complicated. So we'll go 4 minutes. Oh, actually, I'm going to take a picture of this first. Yep. Copy. Okay. So 4 minutes. Here we go. Just a little more time. Just a couple more seconds. Okay. So here's what you should have gotten. We have a female dog that is heterozygous. We have a male dog that is heterozygous. So if they're both heterozygous, that means that we're going to see a lot of stuff with our outcomes. Heterozygous means that we got a capital and a lowercase. So we got both. Similarly, over here, we got a capital and a lowercase. So when it comes time to do your crosses, you bring it down, you bring it over. You're going to have big F, big F, big F, little f, big F, little f, little f, little f. So now it's going to make all of this a little bit more involved. We're seeing then all three genotypes. We're not just seeing one or two anymore. We're seeing all three of them. So big F, there's 1 out of 4. So that's going to be 25%. Now, big F, little f, there's 2 out of 4, which reduces down to 1/2, and that's going to be 50%. Just like you saw with big F, when it comes to the homozygous recessive, little f, we see that there's 1 out of 4, which is going to be 25%. So all three genotypes are present in different percentages, 25, 50, 25. Now, this is often referred to as a 1 to 2 to 1 ratio. The star likes to say that sometimes, so just keep that in mind. Two heterozygotes will always result in a 1 to 2 to 1 ratio. So that means that for every one of these, you have 2 of these and 1 of these. That's all that means. Now, your phenotypic ratio, a little bit different, right? When it comes to black fur, I'm going to make this black. This is black, right? This one. Little f, little f. I guess I should do one more to make it look gray. There we go. Does it look like it's black yet? You know what? That's too much work for not a good result. We'll just go like this. Yep. This is going to be black. Everything else then is going to be gray. So this was a nice gray when I used the highlighter. Ooh. See, this is why Classic is fun. You get to like fill in the blanks. Can you do this on a PDF? Don't think so. Can you do this on a Word doc? Don't think so. At least not as easy as this. Right? So we know that gray is the dominant allele. So all three of these boxes are going to express that dominant allele versus the recessive allele, which is only going to be in that box. So black fur, we're going to see it as, oops, make sure we get this back to no background, and I guess we'll keep red. I like red. Black fur is going to be 1 out of 4, so that's going to be 25%. Now the gray fur is 3 out of 4, and that's going to be 75%. So again, if you wanted to turn this into a math problem, you could say that, you know, if there's a litter of 100 puppies, how many of them would you expect to be black? And the answer would be 25. Okay, that's fun, right? Let me take a picture of it so that we can move on. So Punnett squares, and the thing that we'll do after this when we get back from spring break is called pedigrees, like family trees. I think they're pretty fun. Okay? Now, one more. This time we're dealing with celebrities. We got an A-lister and a C-lister. So if Wentworth Miller has a widow's peak and Rihanna does not, they want to know what their kids are going to look like. So I'm going to give you 3 minutes. Here we go. If you don't know what a widow's peak is You. Oh, let's make this something you could see. How about white? So it goes down to a point like that. Versus Rihanna, see how Rihanna has like nothing there? This one goes down to a point, so that's the widow's peak. If you pull back your hair, you should be able to see it like this. It is. It's Captain Cold. We got a Arrowverse fan. I'm not a big fan of Arrow myself. I like Flash. Flash is good. I dropped off a couple years ago, so now there's all these other shows, right? You got the Batwoman or whatever. You got what's the time travel one called? My brain wants to say Defenders of the Earth, but that's not what it is. Um, Arrow just seemed really dramatic. Yeah, Legends of Tomorrow. There you go. Legends of Tomorrow seems really dorky too. For some reason, I don't mind it with Flash. Flash has got those really soapy elements, but he punches a gorilla sometimes, and sometimes that gorilla punches a big shark. So how could you hate that? You can't hate that. That's great. Yeah, me too. When I'm forced to watch the other shows, and I have no idea what's going on. I can't say that I hate it because I've never actually watched it except for the crossovers. It just looks like something I wouldn't enjoy. It looks like if Batman met, I don't know, Gilmore Girls or something. It just seems really dramatic. About 30 more seconds. My wife hates it. She hates watching Flash. So I try to watch Flash when she's not around. Okay. So let's go ahead with this one. This one's pretty simple. So we got our square. Let me make it black again. Boop. We know that widow's peaks are dominant and no widow's peak is recessive. So we got Rihanna over here. We're going to go with little a for Rihanna. Wentworth Miller is going to be big A, little a. So you go through it, you got big A, little a, big A, little a, little a, little a, little a, and little a. So to make it really quick, it looks like it's 2 out of 2, right? So you got a 50% chance that it's going to be widow's peak, and a 50% chance that it's going to be no widow's peak. Now, why is it called widow's peak? Don't remember. Something about 1600s. It's been a long time. Okay. There we go. Actually, no, I'm going to include our genotypes just for funsies. There we go. Okay. Let me snap a pic of this. Copy. Okay. So that was all the practice we're going to do with Punnett squares. If you don't remember, tomorrow is your mock exam, right? So again, if you need the lockdown browser, make sure you come into campus 4 to 6, so you can download it onto your school issued device. I know that people who have been trying to do it at home are having trouble, and I think even not doing it correctly. So make sure that you are coming to campus to get that done if you are not going to be coming to campus for the actual mock exam. Okay, sickle cell shaped red blood cells result from a mutation in the gene that codes for hemoglobin. This mutation results in sickle cell anemia. A partial sequence of bases formed from a normal hemoglobin gene and sequence that results in sickle cell anemia are shown below. So we did a problem very similar to this. You see the normal sequence, you see the sickle cell sequence. What type of mutation? Right off the bat, you can get rid of 3 of these. Like, you just look at this question and already know the answer without even really looking at it that hard. So let me help you. Look at this. Look at this. Same numbers, right? Nothing is changing. Well, there's something changing in there, but the number of nucleotides is not changing. That means you get rid of insertion. That means you get rid of deletion. That means you can probably get rid of frame shift. I guess you can leave it for now since you're not 100% sure, but you can probably get rid of it because everything else looks the same. So T, G, A, G, G, T, C. Here's our difference. C, C, T, C. So nothing changes after our difference. So it's not a frame shift. So even if you didn't know a whole lot about mutations, you can get to the answer pretty quickly.
Okay, so
Did you have a question?
yes.
Go for it.
So isn't substitution a type of frameshift mutation?
Substitutions can cause frame shifts, but typically they don't. Usually a substitution is just replacing one nucleotide. So here we had a T, it was substituted out with an A, so it did not cause a frame shift. Now, insertions and deletions most definitely will cause frame shifts. So substitutions can, not super common. Yeah, Eileen, I think you're referring to the lockdown browser. Since the test is tomorrow, you need to come to school today at like 4 to 6 to make sure that your computer has it, if you're going to stay home. I think there's a tech guy here who's downloading the app to the Chromebooks, whatever computer y'all have. Okay, okay.
But sure, I thought it was mandatory. So,
I mean, you're supposed to come here, but I know that some people are not going to. I think that's just the nature of things, and I'm not going to force anyone to come to campus because I wouldn't be here if I didn't have to be. But if you are not coming to campus, you still have to have the program on your computer in order to take the test because we're getting the computers ready for the real thing. Because again, the STAR is only going to be in about a month and a half, a little bit less. So it's important to have your computers ready for that. So you have to have that thing installed because it'll be what the STAR runs on later. Okay, a diagram of DNA strand is shown below, arrow pointing to which component of the DNA strand. So we know that this part of the DNA and this part of the DNA are different, right? If we're looking at a nucleotide, there's our 5-carbon sugar, there's our nitrogenous base, and here is our phosphate group. So if that's our nucleotide, let's make it a little bit smaller, a little bit longer. That's going to be your sugar, phosphate, and finally your nitrogenous base. Now remember, the sugar and the phosphate make up the sugar-phosphate backbone, right? That's the stuff that's going to be on the side. The stuff in the middle then, that's going to be your nitrogenous bases, remember? So go to your answer choices. Says, let's see, arrow pointing to what component? So the arrow is pointing to this space. You see the things lining up, right, to attach to each other? So odds are you're dealing with your A's, T's, C's, and G's, right? So we can probably get rid of amino acids, right? We are definitely not talking about proteins. Now, we are dealing with a section of DNA, so it could be talking about genes, but specifically we're looking at something inside of the genes, so you can get rid of that. As I said a second ago, the thing on the outside is going to be the sugar phosphate backbone. This is pointing to the stuff on the inside. So that only leaves you with one answer choice, the nitrogenous bases. And remember, those are held together with a hydrogen bond. Boop. Boop. H bonds. Okay. Easy peasy, right? Nobody missed any of these questions. All right, I see another codon chart. I love these because they're just like puzzles. Let's go ahead and solve it. Which of these changes to the DNA triplet will affect the protein produced? I think the reason why a lot of people missed this question is because you had to do work, right? So GCT, that is your DNA sequence. So we've got a T in there, right? What you have to do then is you have to transcribe it. So G to C, C to G, T to A. That's going to be the mRNA codon that we are then going to translate. So CGA, you go up here. CGA is arginine. So what you're looking for with these, one of these is not going to code for arginine, right? Because it says which of the changes to the DNA will affect the protein produced. So if it's going to affect the protein, it's going to have to be making something that is not the protein, right? So we got to do the same thing here. GTT is going to be CAA. TCT is going to be AGA. TCC is going to be AGG. And GCA is going to be CGU. Let's make it a little bit smaller. That works. Okay, so CAA, that's going to code for CAA, glutamine. So you can probably stop there. But just because you want to be sure you're not making any mistakes like your teacher makes sometimes, you go ahead and do the rest. AGA, arginine, that checks out. AGG, AGG, arginine. And last but not least, CGU, I think we all know what it's going to be, assuming we did it right. CGU, arginine. There you go. So you're comparing this to those. This looks like it's going to be this one, this one, this one, which means that this is your answer because it is coding for a different amino acid. So it's going to affect the protein. Okay, organisms can be classified based on homology, which is shared characteristics inherited from a common ancestor. In the past, homologies were based on studies of anatomical structures and patterns of embryonic development. In more recent years, the use of molecular biology techniques has allowed homologies to be compared at the level of nucleotide sequences. Whoa, we're just given a whole encyclopedia worth of information, right? Next sentence. Nucleotide sequence comparisons are possible because all organisms share which of the following. So here we're told a whole bunch of nonsense about the past and homologies, whatever. The question is saying nucleotide sequences comparisons specifically are possible because they share which of the following. So what is common to all things that are alive? Now we know that organelles are not universal, right? We know that there's a division between eukaryotes and prokaryotes. Because of that, we know that prokaryotes don't necessarily have the same organelles that a eukaryote does. So that's not going to be common across all life, right? Division of the nuclear chromosomes. Whoa, what? I don't even know what that's really talking about. So let's put a question mark there. Boop. Types of proteins needed for cellular functions. Now, it makes sense that if we're talking about all life, right, we're going to be talking about a bunch of different proteins. So that's not necessarily something that's going to be shared across all life, right? All life are going to probably have different proteins. So they're not going to be the same proteins. So the reason we can do comparisons are because all things that are alive are going to have the same DNA bases, right? A, T, C, and G are common to all living things. Yep. Miguel, you were off mute, but I assume it was by an accident. Okay, last but not least, our transcription translation problem. We're shown a diagram here. You can see transcription happening up here, DNA to RNA, and then translation to protein, right? So remember that transcription is making the mRNA copy, right? That's the whole point of transcription. Translation then is taking that mRNA copy and turning it into the protein it codes for. So let's go ahead and say DNA here. Right, so this is what's going on. We see that there's transcription, there's translation. We are very familiar with these processes, so let's get down to the question. What statement correctly describes part of the process in the diagram above? So something about growing protein has amino acids in the sequence coded by the cytoplasm. 2 subunits of the ribosomes translate the DNA code into mRNA codons. DNA is transcribed into ribosomes to form proteins in the cytoplasm. Anticodons on tRNA pair with mRNA codons to join amino acids to form proteins. So, where do we start? Which one of these do we get rid of? Uh-oh, I think you're stumped. And I think that was reflected in the test a little bit. Can anybody let me know which one of these F, G, H, or J can we get rid of?
F,
Why?
because it's talking about the protein, but it's not saying like how it got there. It's just saying it's a growing protein. Like,
That's true. It's also really important to make sure that you're reading the question, right? We're looking for the one that's correctly describing the process. So if you're looking at 24, it says growing protein has amino acids. That sounds right. Coded by the cytoplasm. We know that the proteins are coded for by the DNA, right? Not the cytoplasm. So that's not right. Skip down to the next one. The 2 subunits of the ribosome translate the DNA code into mRNA codons. Right off the bat, it sounds a little bit fishy. Translating, I think, usually means mRNA to protein, right? But we're not sure because it mentions enough words that you think it could be true. So let's skip that one. DNA is transcribed into ribosomes to form proteins in the cytoplasm. Another head scratcher. Kind of sounds true. Something about DNA and ribosomes, proteins, all that sounds okay. It doesn't sound as wrong as that first one. So let's go to the last one. Anticodons on tRNA pair with mRNA codons to join amino acids to form proteins. Now, this one sounds very accurate when you're comparing the other answer choices, correct? So we look here, the 2 subunits of the ribosome translate the DNA code into mRNA codons. We know that the ribosome is translating the mRNA to make proteins. So you get rid of this one. Now we're just left with H and J, which both kind of sounded okay. DNA is transcribed into ribosomes. Remember that DNA is transcribed into mRNA. right? So you get rid of that one. J then is your answer. Now, anticodons on tRNA pair with the mRNA codons to join amino acids to form proteins. Now, this is a difficult question. This is also a star question. So it requires really, really in-depth understanding of how these processes work because you have to weed through the incorrect statements. So that's why I put this over here to the left, just to remind you of what these processes are doing, right? That's really what the question is getting at, is how well do you understand the differences between transcription and translation? And the best way to figure that out is by shoving a bunch of incorrect information at you and you having to figure out which one of those is the correct option. So it's a difficult question, and a lot of y'all missed it, and I think it's because it required a lot of thinking. And secondly, some I think missed it because they didn't read the correctly part here, because we're looking for which statement correctly describes. So only one of those was accurate out of the 4. Okay, we got about, what, 15 minutes left? Go ahead and use this time now for your exit ticket. And I'm going to go ahead and stop recording so that I can put some music on.